Hi everyone, it's Dr. Hall. We're going to continue on our unit with vision. So this is part 2, lecture 10, where we're kind of going into the neurophysiology. So now that we've already talked about the structures of the eye that then direct light rays back onto the retina, what happens next? So this lecture is divided into 3 parts. So first we're going to talk about the cells of the retina, including the photoreceptors, whom you've already met briefly. Then we're going to talk about the neural pathways and kind of the nerve circuits for vision through the brain. And then we're finally going to talk about visual perception, which is different from sensation. So let's get started with retinal cells, 10A. There are 5 layers of retinal cells. So it is 5 layers deep, and let me introduce you to some of these cells. So you have already met down here the photoreceptors, so the rods and the cones. They are actually the deepest layer of the retina, and they are the ones, of course, that are going to sense light. Then we have some cells in yellow here, these little horizontal cells, and they're going to modify signals. You can see how they have connections to lots of different photoreceptors, so they're kind of keeping an eye and modifying things. And then we have the bipolar cells. So the bipolar cells are going to conduct signals that are coming from the photoreceptors, then on to the next layer. As they do so, we'll see these amacrine cells. Again, these are similar to the horizontal cells in that one cell will have lots of connections with different bipolar cells on different levels of the next cell. And so again, they are modifying things that are happening. And then ultimately, we're going to arrive at the retinal ganglion cells. And these are the cells that are actually going to send an action potential out along their axons, which bundled together make up the optic nerve. So again, I just want to reiterate, it doesn't seem to make sense, but the photoreceptors are actually deepest. The other cells are kind of on top of them. So the light rays have to penetrate through in order to reach the photoreceptors. Interesting fact, when we get to the fovea, which remember is that very center of your field of vision, that thing you're looking directly at where you have lots of cones and you have really, really specific detailed, crisp images. The other cells kind of like lean away so that they are less in the way. So they're kind of out diagonally like this. So when you actually look at the fovea on someone's retina, it's like a little pit because those cells that are normally, right, are just kind of leaning away. Anyway, it's weird, but there we go. So how do photoreceptors work? So whether you are a rod cell or a cone cell, the basic principle is the same. You have certain photopigment molecules inside the upper part, which is actually kind of the deepest part of the cell, that are molecules that are going to actually change in their shape and their conformation in response to light. So when they're hit by certain wavelengths of light, those molecules are going to change. Changes in those molecules then set off a whole sequence of events that lead to the opening or closing of ion channels. And so ions are going to flow according to their electrochemical gradient. And so depending on what happens, the cell can either depolarize or hyperpolarize. So you might be thinking, oh, and then is it going to send an action potential? No. So photoreceptors are strange in that they don't send action potentials, but what they will do is, depending on what's happening with their membrane potential, they will release a neurotransmitter down from their synaptic terminal, and they release a neurotransmitter called glutamate. And then that glutamate is going to send a signal to other cells in those retinal layers. So kind of weird, kind of interesting, no action potentials, just relative changes in membrane potential that cause you to release more or less glutamate into the next synapse. And it turns out, we kind of think of like, okay, if you're stimulated by light, then you're going to release glutamate, but actually no. So when there is no light hitting the photoreceptor, as we can see in this diagram, so it's dark. So when there is no light, you are releasing glutamate. So here we are in the terminal. right? Here's our little glutamate synaptic vesicles, and we're releasing glutamate into the synapse. Let me pick a different color. We're releasing glutamate. Didn't change my color. Try again. It's releasing glutamate into the synapse when it's dark. Now you might notice there's this extra yellow cell. That's one of those horizontal cells that's modifying. You might say to yourself, that looks like a tripartite synapse, like we talked about with neuroglia way back in the beginning part of this semester. Yeah, it's similar, but different. Okay, so in the dark, photoreceptors release glutamate. And then when they are stimulated by whichever wavelength of light they respond to, depending on if they're a rod or a cone and which color cone, then they release less. So you can see under the light conditions here, there is much less neurotransmitter being released to the next cell downstream, which is a bipolar cell. So it's kind of interesting that these cells don't use action potentials. Instead, they release glutamate in the absence of light stimulus, and they release less glutamate when they are stimulated by their specific frequencies of light. Okay, so we talked about how the photoreceptors are going to send a signal or not. So let's kind of put it all together. We've got some light coming in to the retina. So remember, the photoreceptors are the deepest layer. But I put light down there so that we can be like, okay, light reaches them. And so they're going to release less glutamate into the synapse to the bipolar cells. We're going to leave out the horizontal cells and the amacrine cells just to focus on the direct signal transduction or movement of the signal from one cell to another. So they're going to release less glutamate, which is then going to signal the bipolar cells. Now, bipolar cells come in different varieties and they're going to respond differently. So we're going to use generic terms here. And then the bipolar cells themselves release more or less glutamate depending on what kind of signal they got from the photoreceptors. And then depending on what the bipolar cells released, the retinal ganglion cells here on the top are going to send an action potential out their axons, and their axons, of course, then bundle together to form the optic nerve. So that vision signal, right, is, get my little, let's try purple. Okay, so here is this section of retina. So as we look at it here, that's those 5 layers deep, and they're just showing you the rods and the cones in pink and yellow. And then those signals move through the bipolar cells up to the retinal ganglion cells here. Right, so moving up from here to there, and then down the optic nerve to the brain. Couple of things. One is this concept of synaptic convergence. So it just so happens that every cone cell has a one-to-one relationship. So every cone cell here, you see this here, let me change to red. Every cone cell synapses with one bipolar cell, which then synapses with one retinal ganglion cell, which then sends its individual axon through the optic nerve. This gives us very, very high resolution. It's a one-to-one relationship, whereas rods often have to kind of share. So here we've got, how many have we got? We've got 9 rod cells, right? And 3 of them have to share this bipolar cell, right? And then there's 3 of these bipolar cells to signal this one retinal ganglion cell. So if this one retinal ganglion cell fires, you don't know which of the 3 bipolar cells, or certainly not which of the 9 rod cells it was that was actually activated by light. So this is less specific. It's more fuzzy. You don't really know exactly where it came from, but it also allows for you to perceive light in low light conditions, right? Because if any one of these perceives light, it will send the signal on. So pros and cons, that's why we use these 2 different photoreceptor types. Oh, look, forgot. I put that in for you to orient you. There we go. Okay, this is showing you the same thing, just kind of in a flipped different way. So what they're showing us is cell number 1 is located at the fovea. And so cell number 1 is a cone cell. So here is cell number 1 right here. It's a cone cell. And so it's got this one-to-one relationship with a retinal ganglion cell. So when that retinal ganglion cell sends its action potential, we know exactly which part of the retina we're talking about. If we look at retinal ganglion cell number 2, which is a little bit more out toward the periphery, we can see that its setup here is it's receiving potential input from 4 different rod cells. So that's covering a bigger area of the retina. So when this retinal ganglion cell fires, we know it was somewhere in this area, right? But it's much less precise. And then if we look at cell 3, which is even farther out in the periphery, okay, this is the one with 9, right? So this has a very large, what we would call receptive field, meaning it could have come from any one of these guys, so we don't know exactly. We don't have precise information. You can think of it a little bit like a one-to-one text conversation, where if you hear your phone ping, you know exactly who it's from, versus if you're in one of those group texts and you hear your phone ping, you don't know, like, was it Jeff or was it George or was it Sally, right? You don't know exactly who it was from. So less specific. So retinal cells, in summary, 5 layers, lots of cells. We've got those photoreceptors, our rods and our cones, and in response to light, that photopigment is going to change, that molecule is going to change, causes a sequence of events to happen inside the cell, changing the membrane potential, which then will lead to a change in how much glutamate it releases from its synapse. Horizontal cells are modulating. Bipolar cells then are synapsing. They're the postsynaptic cell for the photoreceptor cells, and they're going to respond to that glutamate release from the photoreceptor cells by either releasing more or less glutamate themselves. And then amacrine cells are modulating. Ultimately, then we get to the retinal ganglion cells that, depending on what kind of glutamate they're getting from the bipolar cells, if they depolarize, right, if they reach that membrane threshold, they will fire an action potential, do all the things we learned about at the beginning of the course with the sodium channels opening, sodium rushes in down the axon, which is, of course, traveling within the optic nerve. Then we also talked about synaptic convergence, that fact that cone cells get a one-to-one relationship with retinal ganglion cells. So you always know exactly where on the retina that light is coming from. But with rods, it could be from a variety of different ones. So we just get this kind of fuzzy, imprecise picture. All right, in 10B, we'll talk about neural pathways now. Once we're in the optic nerve, where are these signals going? See you there.